Hello and welcome to chapter 4.3, Transport Across the Cell Surface Membrane. Okay, so a little bit of this is going to be review and from the last few slides, and some of this will be new stuff for you, so let's get started. We've been talking about the phospholipid bilayer and how it creates this effective barrier against water-soluble molecules and ions. It prevents aqueous contents from escaping, and some essential transport is achieved through diffusion, facilitated diffusion, osmosis, active transport, and or bulk transport, and we've covered those. We're going to talk a little bit of detail about those. So diffusion, as we've talked about, is the net movement as a result of random motion of the molecules or ions, and those molecules are always going to randomly, right, over time, move from a higher concentration to a region of lower concentration. And in this way, particles move down a concentration gradient. Okay, so the steepness of concentration gradient is the greater the difference equals the higher the rate. Okay, and temperature, higher temperature equals a higher rate. Surface area of diffusion, the greater the surface area, the less the rate. The higher the rate. And the nature of molecules and ions are small molecules equals a greater rate. Okay, so again, these are the things that affect rate of diffusion: the steepness of the concentration gradient, the temperature, the surface area, and the nature of the molecules. Okay, so respiratory gases cross the membrane by diffusion, and this is an uncharged and nonpolar. And water, even though it is polar, can cross because the molecules are very tiny. Okay, so again, I encourage you to pause this video and look at all of these different models that I have here for you. Okay, so here, like you can see, the hydrophobic molecules, small uncharged polar molecules have an easier time getting right through and diffusing through that layer than large uncharged polar molecules or ions. Okay, diffusion that takes place with the help of a certain protein molecule, like a channel protein or a carrier protein, is called facilitated diffusion. And channel proteins, these are water-filled pores that have a fixed shape. And in this way, it allows charged substances, obviously usually ions, to diffuse through the membrane. And they are gated, okay? And this is to allow for selectivity and to control movement. Again, pause these videos and really look at these images that I have here. Okay, carrier proteins. So flips between shapes to alternately open binding sites on interior-exterior membrane. So the direction of movement will depend on the concentration gradient inside and outside of the cell. So it's in that way they're gated, like we were talking about before. It will flip shapes, and I want you to keep in mind that these are models of the way that we understand how these things work. And so when I say flips between shapes, really it's one side will open on one side of the cell and then the other will open on the other side of the cell, and it's really about a shape changing, allowing the protein to open and allow a solute in. Okay, so osmosis is the type of diffusion that only involves water molecules. Okay, and it's across a selectively permeable membrane. Or not selectively, it's across a permeable membrane. So let's talk about water potential. Water potential is a tendency of water molecules to move from one place to another. Okay, and it's measured by the Greek symbol psi. Water potential for pure water is 0. Solutes make water potential less than 0. The more solute, the more negative the water potential. The amount that solutes lower the water potential is called the solute potential. So water will always move from a high potential to a low potential, okay? You can think about it the same as the concentration gradient, right? Water will move from a high potential to a low potential. And when you add solute, so when you're looking at this diagram, when you have pure water on one side, at one time that whole thing's pure water. When you add solute to the part on the right, it is increasing the water potential, right? So then the water will move through random movement, will diffuse down the concentration gradient to the lower water potential. Okay, so water potentials are always from a high to a low. So it's going to be the same. If you look at the one on the right, you have a plant cell that's inside pure water, okay? And the water potential on the inside of the cell is higher than the water potential on the outside of the cell. Okay, pressure potential. So increasing pressure will increase water potential. The contribution made by pressure to water potential is called pressure potential and is given by a symbol psi sub p. Pressure potential makes water potential less negative. So as water is putting pressure on this cell in the image here, and the pressure inside of the cell is becoming less negative. Water potential in animal cells. So cells are placed in distilled water. The cell can swell and burst, and vice versa. If cells are placed in concentrated salt solution, the cells can shrink and shrivel. So osmosis in plant cells. So this is when you're thinking about a plant cell inside pure water or any type of solution, you're still dealing with the osmotic pressure. So if you're looking at a hypotonic solution is a solution in which the water or the solution itself has a lower concentration of solutes than that of the cell. Okay, and so in these conditions, the cell is going to be turgid or normal. It's going to have water inside of the cell, and there's water that's outside of the cell. Water will come into the cell from the solution. However, it is not going to burst the cell because of that cell wall, and it's in that hypotonic solution. So moving on to isotonic solution. An isotonic solution is when the concentration of solutes is the same in the solution as in the cell itself. And in this case, water is going to move in and out of the cell, but the cell itself is flaccid. Okay, and so this is kind of like inelastic. Okay, it's an inelastic consistency, and it's really the case because water is coming out of the cell. It's moving in, but it's also moving out, so it has this kind of flaccid shape. And then in a hypertonic solution, this is where there's a greater concentration of solutes, and this is where water is moving outside of the cell into the surrounding water because, again, the concentration of solute in the water is greater than that in the cell. So water is moving down the concentration gradient from inside the cell to the solution. Okay, so I'd like you to draw a membrane and label what will keep out ions, and as well what will increase membrane fluidity, what will control rigidity and help keep out polar molecules, also what allows for cell recognition and what allows for polar molecules to enter. So pause the video right now and you can turn that in on Schoology when you're done. Okay, so let's talk about active transport. Active transport moves molecules or ions against the concentration gradient. So we've been talking about passive transport and the different type of molecules that achieve that, but in active transport, energy is going to be needed to move things against a concentration gradient. This is achieved by carrier proteins, and this is specific for the structure of the particular ion that is being moved. It requires energy that is supplied through cellular respiration, and it can occur moving things out of the cell or into the cell. So again, the key here with active transport is moving things against the concentration gradient. Okay, so one example of this is the sodium-potassium pump. So this is found in the cell membrane of all animals. It uses 30% of the average cell energy and 70% of the neuron cell energy to achieve this type of movement. So pump 3 sodium ions out of the cell and 2 sodium plus ions into the cell. Okay, so 3 sodium ions and 2 sodium ions. The net result is that the inside of the cell becomes more negative than the outside. And so I'd like you to pause this video and really take a look at these images here. Okay, the difference in charges between the inside membrane and the outside creates a potential difference. And this allows nerve cells to propagate a signal. Okay, we can't, so otherwise we'd be dealing in a net neutral environment, and in order for neurons to move charge, there has to be a situation where there is more of a charge on one side of a membrane than the other, which is called a potential difference. Okay, it's also important in reabsorption in the kidneys, where certain useful molecules and ions have to be reabsorbed into the blood after filtration into the kidney tubules. It's involved in the absorption of products of digestion into the gut as well. It's used to load sugar from photosynthesizing cells of leaves into the phloem tissue for transport around the plant. Okay, and again, these are examples that Cambridge is going to want you to know. These are specific examples from the textbook. All of these images you should know by heart, and each of these examples should be a way that you can explain how active transport is important in our bodies and in the bodies of plants.
A cell may need to have a substance cross the plasma membrane against its concentration gradient. This requires energy and is accomplished by active transport. The sodium-potassium pump is an example of this process. A large protein in the plasma membrane provides the doorway through which sodium and potassium ions can move. ATP is the energy source. Sodium inside the cell binds to the protein. The addition of a phosphate group of ATP changes the shape of the protein, and the sodium is expelled. Now potassium binds to the protein. The phosphate is released, and as the protein returns to its former shape, potassium is moved into the cell. Sodium can once again bind to the protein, and the process repeats as long as there is a supply of ATP.
Okay, so bulk transport involves the mechanism of moving large quantities of molecules into the cell, endocytosis, or out of the cell, which again is exocytosis. Large molecules like proteins or polysaccharides, parts of cells, and even whole cells can be transported across a membrane. So this involves engulfing the material by the cell membrane to form a sac, okay, called an endocytic vacuole. So you should be used to the idea now that a vacuole means a sac that has something in it, okay? So an endocytic vacuole is something where something is moving from the outside of the cell inside, okay? So there are two forms. There's phagocytosis and penocytosis. Okay, phagocytosis is, in other words, is cell eating. Okay, this is bulk uptake of solid materials. The cells specialize in this are called phagocytes, and the vacuoles are called phagocytic vacuoles. Okay, so here's an example of the engulfing of bacteria by immune cells. Okay, again, pause these videos and look at every single image. So here you can see a phagocyte chasing a cell and engulfing it. Okay, so penocytosis is known as cell drinking, and this is bulk uptake of a liquid into a cell. Okay, phagocytosis is for solids, penocytosis is for liquids. So these vacuoles are formed in a very small form. And the process is called micropinocytosis. So human egg cells that take up nutrients from cells that surround it by pinocytosis. So look, if you really look at this model here, I'd like you to pause the video and really look at this model. Remember, extracellular fluid is anything that is outside of the cell. Remember, any example, again, any example that I give you is an example that Cambridge has given you. And so I really need you to look at all of these images and make sure that you're memorizing these examples. So again, an example for pinocytosis is, this is how a human egg cell will take nutrients from its surroundings. Okay, so exocytosis, this is the process by which bulk material is removed from a cell. Up until this point, we've been talking about endocytosis, which is taking things into the cell. So an example of this are how plants transport materials out of the cell membrane to make a cell wall, or the secretion of digestive enzymes in the pancreas, right? So a secretory vesicle from the Golgi will carry enzymes to the cell membrane in order to release the contents out of the cell. Okay, that's it for exocytosis. See you in class.